Hello everyone, my name is Rachita and today I will be presenting a research article titled Altering the Substrate Specificity of Acetyl-CoA Synthetase by Rational Mutagenesis of the Carboxylate Binding Pocket by Sophil and the colleagues, published in ACS Synthetic Biology in 2019. This study investigates whether the scientist can redesign an enzyme so that it can recognize different substrates than it naturally does. Specifically, the researchers attempted to modify acetyl-CoA synthetase or ACS to utilize larger carboxylate molecules. So let's go into the background so that we know the background before we get into the real study. So as we can see that acetyl-CoA synthetase or ACS converts into acetyl-CoA. Acetyl-CoA is one of the most important molecules in the metabolism because it participates in the TCA cycle, which is fatty acid biosynthesis, like in fatty acid biosynthesis, amino acid metabolism, and many other cellular processes. Naturally, ACS strongly prefers acetate, which is a 2-carbon molecule. The researchers wanted to determine whether this preference could be changed through protein engineering. So, the main problem here is that the wild-type ACS is highly specific for acetate and cannot efficiently use larger carboxylates. Therefore, the research question was, can ACS be redesigned to utilize larger straight-chain and branched-chain carboxylates? The researchers hypothesized that changing amino acids within the substrate binding pocket would alter the substrate specificity. So, what research roadmap they actually followed? I tried to summarize the overall workflow. The researcher started with the wild-type ACS and used computational structural modeling to identify important residues within the binding pocket. Then they generated mutant enzyme using site-directed mutagenesis. Then the mutants were tested against the multiple substrates, and their activities were compared to determine how substrates specifically changed. So, how ACS actually work? So here you can see in the step 1, it actually works in the 2 steps. It's like ACS catalyzes a 2-step reaction. First, acetylate react with ATP from an acetyl AMP intermediate. Second, the coenzyme A reacts with this intermediate to produce acetyl-CoA. So the researchers were not trying to change this reaction mechanism actually. Instead, they wanted to change which substrates could enter the binding pocket and participate in the reaction. So finding the important residues are to identify the residues involved in the substrate recognition. The researchers compared Arabidopsis ACS with a related enzyme from Pseudomonas that naturally uses a larger substrate. Using sequence alignment, the homology modeling, the I-Tessor structural prediction, they searched for differences in the binding pockets. So their goal was to identify residues that in the identification process of the binding pocket, the structural modeling identified 4 important residues. They were predicted from the carboxylate binding pockets because the substrates must fit into the pocket before the reaction occurs. Actually, the researcher predicted that the changing these amino acids would alter the substrate preference. These 4 residues became the targets for mutagenesis. And you can see those 4 key residues here. So what the experimental design they designed, the researchers actually created a single mutant, double mutant, and triple mutant. So each enzyme variant was tested against several straight chains and branch chain carboxylates. By measuring enzyme activity, they could determine how mutation affected the substrate specificity. So in the results, what they got is the first mutation replaced threonine with the triosine. This mutation completely eliminated the enzyme activity. As you can see, the triosine is much larger than threonine, causing the binding pocket become smaller. As a result, substrate could not properly address the active site. This experiment showed that the THR324 is essential for maintaining a functional binding pocket. So in this slide, we can see that the TRP 427GLY mutation produced one of the most important findings, replacing the large tryptophan residue with the much smaller glycine expand the binding pocket. As a result, the enzyme gained active larger chain substrates, including byrate, valerate, hexonate, and hepanooctanoate. This demonstrated that binding pocket size plays a major role in the substrate specificity. So in the results in here, the researcher combined this vol and drop compared with the wild type enzyme activity towards the acetate decreased while the activity towards larger substrate increased. Most importantly, the butyrate became the preferred substrate. This showed that the combining mutation can shift substrate preference away from the natural substrate. So what is this results for triple mutant? This is the most important results of the study. The triple mutant combined the THR324 gly and vol 399 ella and TRP 427 gly. This variant showed strong activity towards hexonate and several branch chain carboxylates. Compared with the wild type enzyme, substrate specificity was dramatically altered. The researchers successfully redirected the enzyme activity towards the larger substrates. So why this mutation worked? Actually, the researcher concluded those two factors control substrate specifically. The first is the binding pocket size. The larger pockets allow larger substrates to enter. The second binding pocket is the chemistry itself, because it changes in the amino acids and composition alter how substrates interact with the enzyme itself. Together, this changes explain that the altered substrate preferences observed in the mutants. So in the summary of the mutants, I would say that like this wild type enzyme is a preferred acetate. The TRP 427 gly explained activity towards the larger substrates. The double mutant shifted preference towards the butyrate. Finally, the triple mutant efficiently utilized the hexonate and branched chain carboxylates. This demonstrates how rational mutagenesis is progressively changed in enzyme function. So in the significance and application, this study has important application in the synthetic biology and metabolic engineering. The engineered enzyme could potentially be used to produce biofuels, fatty acid, and polyketides, and other valuable industrial compounds. The study demonstrates how structure-guided protein engineering can create enzyme with new catalytic capabilities. So this one strength of the study is a combination of the computational modeling and experimental validation. The researcher developed a clear hypothesis and systematically tested it. One limitation is that the mutation structure were predicted computationally rather than experimentally determined. In addition, like most experiments were performed in vitro rather than living organism. That's the one limitation that we can see in here. So, at the last, in the conclusion, I will say that the researchers successfully altered the ACS substrate specificity through the rational mutagenesis residues involved in the substrate recognition and demonstrated that modifying these residues can dramatically change the enzyme activity. The most successful mutant redirected the ACS towards the larger straight chain and branch chain carboxylate. Overall, I think this study highlights the power of the structure guided enzyme and engineering its potential application in biotechnology and synthetic biology. And I would thank you for your attention, and I would be very happy to answer any question that you ask in the discussion. Thank you very much.